So the interruption of blood flow to crucial organs like the heart and the brain can lead to sudden death. And this is currently the leading cause of death in the modern world. In the US alone, every 30 seconds, one person dies. But what's more concerning about this is that many of these events occur without any prior symptoms or any prior warning. So I'm going to spend a few minutes and just talk about what excites me about this, of trying to figure out can computational models and supercomputing make an impact and give doctors more predictive and preventative power. This starts with modeling, creating a computational model of the body. So the circulatory system is a complex maze of vessels that's running from the heart to the rest of the body, carrying blood with its nutrients and its oxygen to the other organs. Now, the blood is subject to the laws of fluid dynamics, which is greatly influenced by the components of the blood, like the red blood cells, the white blood cells, and specifically the geometry of the vasculature that the blood is moving through. Heart disease develops over the course of months to years, and in this case, you're going to see, you would see plaque buildup in the walls, typically in areas where our collaborators have identified that the plaque tends to build up in regions where there's low shear stress at the walls and the arteries. And this buildup of plaque can lead to blockage of the flow and potentially a rupture, which then leads to these cardiac events. The problem is we can't actually measure where the regions of low shear stress are in the body. So that's where simulations like the ones I'm going to show you can actually come in and play a role to identify in a patient-specific manner where these regions of low shear stress are. So we're getting data from patient-specific images. So we're getting data from CT scans, from MRI scans, to create an image-based patient model of the arterial tree. So we're segmenting this data from the CT scans and creating patient models of the vasculature. From there, we can apply a three-dimensional Cartesian grid across the mesh of the vessels. And at each point in the grid, we identify if it's a fluid node, an inlet node, wall node, et cetera. And at these points, we can solve equations of fluid dynamics and get a feel for what's going on in the system. And we also model the movement of the red blood cells through these images. Now, the complex geometries involved in the number of red blood cells requires large computational power because you're modeling on the order of 200 to 300 million red blood cells simultaneously just for the coronary arteries themselves, and using billions of fluid points for these grid sections. So we're using some of the biggest supercomputers in the world. Typically, we're using the ones over at their IBM Blue Gene Qs out at Lawrence Livermore National Lab in California. And usually the systems are being used for nuclear stockpile stewardship and weapons modeling, but we're repurposing them to do cardiovascular models. And so we're using probably some of the top, within the top 5 fastest and most powerful supercomputers to get some of these models. And this is the type of output that you're getting. So we can identify the velocity streamlines, get a flow profile, as well as this risk profile that you're seeing on the far side. So that everything on the far side that's colored in the red, that you can see kind of at the junctions there, is where you're really going to be at high risk for that particular patient to develop heart disease. So we can feed that back to the physicians and help them identify where these regions are. So the video that had played a few seconds ago was the model of one cardiac cycle, and to get that completed, it required 6 full hours on a supercomputer that contained 200,000 processors. So it kind of gives you an idea for the order of magnitude of the simulations at this point in time. And the goal is, as technology develops, we can then bring this down. So this is something that physicians could actually do on a real-time basis in the office. And just a quick sentence, if we can, yeah. That's great. Thank you.
Well, thank you for putting up with the glitches that were in that then. But I'm trying to get my mind around the idea that something that at the moment requires a significant fraction of the entire country's computing capacity, as it seems,
yes,
could, in the foreseeable future, be put in the hands of physicians to help predict exactly where in the heart or problems are going to arise. Is that what you're aiming to do?
Right. So we're kind of relying on two pieces, where one, as technology is developing, we all know that what you have in your cell phone is similar to what supercomputers were, 10, 20 years ago. So we're relying on that fact, as well as we're understanding and putting as much into the models as we can now, we can figure out what we can pare out and take out to make the model simpler for the future.
Right. And did I understand, I can't resist asking this, that the name you give to this stupendous computing capacity is Harvey?
Yes.
William Harvey?
Yes.
Wonderful. Please.
Hey, I guess I have sort of two questions. How much actual input data do you need? Like, how much time do you have to spend actually recording someone through one of those imaging devices? And then what level of certainty do you actually get out of that? How accurately does it predict this?
So right now, we're getting data that's already been retrieved from a normal CT scan or MRI scan that the patient would be going through. So you can make it as patient-specific as you'd like. So if you have measurements for what their hematocrit levels, like the number of red blood cells they have in the bloodstream, the inflow rates for going into the coronary arteries, whatever measurements you have, we can feed that into the model, but can also use patient-specific or patient average data as well, so we can see what's really necessary. So I'm trying to think of how much. Yeah, it's. Right now, we have it's already tests that are routine, and the patients are going through to begin with. So,
how accurate is it? Like, how well does it actually anticipate? Are you right 90% of the time you anticipate that a heart attack is going to happen, or?
So right now, we're validating against measuring the velocities properly, the pressure measurement properly, and we're within just a few percent. If one of the tests we did was comparing against measurements taken in the body for pressure gradients in the aorta, and we were within about 2% for measuring the pressure. So it's more. We haven't done any long-scale predictive studies yet, but that's in the works.
Well, thank you. Over here, please.
This is really fascinating. I actually also work on blood. So the question is, with sickle cell disease, so that your flow dynamics are completely different. Or when you think of a patient like with anemia,
right?
Have you actually tried to put that in the model, or you need to start with the basic right now?
So actually, recently, we just now can use deformable cells, so you can change the tolerability of the red blood cells. And so we haven't done it yet, but other groups that do similar research have worked specifically on sickle cell, where you can put in the geometries and the rigidity and the differences you'd see with sickle cell. So you can make those cell-specific changes.
Sounds like you two should get together and do some work. Yeah, please.
So as you were talking about the shearing stresses and that being the places where the shearing stresses are lowest, that being where the plaque buildup is, the connection that I was making in my head, since part of my curriculum is earth sciences, is river meanders, and the way that the outer bank is the part that gets cut off because it's the largest circumference and the fastest velocities, but the inner bank gets the deposition. I'm trying to just wrap my head around, is it essentially the same fluid dynamics process that you're talking about?
Pretty much. It's really similar.
So what's the cardiac equivalent of an oxbow lake? That's what you're asking me. Yeah.
It is so you typically.
I assume you just get a blockage instead of an oxbow lake, right?
Right.
Right.
Yeah,
It
essentially.
can't keep meandering out on the outer side. It just removes the plaque.
It's the geometries of the vessels. It's very similar, where you're seeing on the it's the shear stresses.
It's okay, don't worry.
The shear stresses are on the lower shear ends up typically being on the inside wall, where you're seeing the bends. It's typically at the bifurcations in the arteries, but it is also influenced by the regions down the distal to the bifurcations. You can't just say it's every bifurcation. You have to really include the larger vasculature.
Thank you. Please.
That was very good. Question is, how many years before this becomes a mainstream tool in a doctor's office?
I think on an everyday basis, it'll be a little while, but at this point, there are already some FDA-approved simulations, not including red blood cells in that scale, but just looking at the bulk fluid and measuring pressure gradients for some diseases. So it's starting to be used, but it's still a few years off before it's going to be regular practice.
And are you relying exclusively on the increasing power of computers, or is it possible your models will start to rely on less data? Both.
Because for some of the questions we're looking at, you may not need to include the red blood cells, or you may not. We're trying to put as much in as possible now to take it out and see what we can remove to simplify the models and still get the right answer later.
Fascinating. Thank you very much indeed, Amanda. Well.